Imagine if scientists could tell whether a plant is going to be disease-resistant or produce a massive yield without even having to grow it first. It sounds absolutely mind-blowing, right? Well, today in this explainer, that's exactly what we're going to unpack. We're breaking down the incredible, almost magical science of molecular markers. Okay, let's dive into this. How on earth can scientists look at a tiny little seed and predict exactly what kind of plant it's going to become? The secret lies in molecular markers. These microscopic tools allow geneticists to identify crucial traits, things like drought tolerance or disease resistance, at super early stages. It's a literal game changer for agriculture. Today, we're cracking open the geneticist's molecular toolkit. We'll be walking through 1. The magic of markers, 2. How the process actually works, 3. Comparing the different marker types, 4. Essential lab applications, 5. Real-world crop successes, and finally, 6. The future of genetics. Let's kick things off with part 1, the magic of markers. To put it simply, molecular markers are specific, easily identifiable DNA sequences that point out the genetic differences between organisms. Let's say you have 2 wheat plants. One is drought tolerant and the other just isn't. By comparing their DNA, scientists can zero in on the exact genetic region that's directly linked to that drought tolerance. That distinct, unique region, that's detected using a molecular marker. Let's think about this with a really great analogy. You can picture these markers as highway milestones for DNA. Just like a milestone gives you a specific location and direction on a long, winding road, molecular markers help scientists navigate through an organism's massive, incredibly complex DNA sequence to locate a very specific gene or trait. Moving right along to part 2, how the process works. This entire laboratory process basically breaks down into 4 clear steps. Step 1 is DNA extraction, where the DNA is safely removed from the plant. Step 2 is marker detection, where we analyze specific DNA regions using PCR or restriction enzymes. Step 3 involves gel electrophoresis to create band pattern formation, which is essentially visualizing those tiny DNA fragments as physical lines. And finally, step 4 is the comparison of those band patterns. If the final band patterns of our plants look totally different, that means a genetic variation is present. And that variation is exactly what scientists then harness for breeding. Let's jump into part 3, comparing the marker types. Now, what's really interesting about this is our comparative cheat sheet for the toolkit. We basically have 5 major tools available in the lab today: RFLP, RAPID, AFLP, SSR, and SNP. As we look across their techniques, the pros and the cons, you'll notice how every single tool comes with a very specific trade-off between technique, speed, and accuracy. Some tools are going to give you blazing speed, but you lose some accuracy, while others offer incredible precision, but cost way more money and time. Let's break down these laboratory rivalries. Let's pit 2 major techniques head-to-head. On one side, we have RFLP. This is the oldest marker technique in the book. It uses restriction enzymes to cut the DNA and is highly reliable, giving us really detailed genetic info. But the catch? It's incredibly time-consuming and expensive. On the flip side, we have RAPD. This one uses random primers along with PCR. It is wonderfully simple, super cheap, and fast. The downside here is that it suffers from low reproducibility. It's basically the classic battle of slow and steady versus fast but inconsistent. Next up in the ring, it's SSR against SNP. SSR markers look for short, repeated DNA sequences. Think repeating letters like AT. They are widely used and highly informative, though developing their initial primers can definitely be costly. But then we have SNP, and this is where things get really thrilling. SNPs look for just one single base pair change in the entire massive DNA sequence, literally just one A changing to a G. Because of this microscopic single-letter focus, it is extremely accurate and allows for massive genome-wide analysis. The only real downside is that it demands some seriously advanced tech to pull off. And that brings us to our 5th tool, AFLP. This one aims to be the best of both worlds. It actually combines the rock-solid reliability of RFLP with the rapid speed of PCR. The result is a highly polymorphic and fiercely accurate tool. But a quick word of warning to the scientists out there, successfully pulling this off in the lab requires some serious technical skill. All right, moving to part 4, essential lab applications. The practical applications of these tools are absolutely game-changing. Using the comparative toolkit we just talked about, like those super accurate SNPs or informative SSRs, scientists can perform plant breeding, genetic diversity analysis, gene mapping, and even disease diagnosis at lightning speed. But the absolute standout here is marker-assisted selection, or MAS.
Instead of waiting an entire growing season just to see if a plant survives a drought, scientists can make their selections directly at the DNA level on day one. It completely bypasses all those messy environmental effects. Of course, using this tech comes with its own set of trade-offs. The upside is massive. It saves huge amounts of time, enables early-stage selection, is phenomenally accurate, and stays completely unaffected by environmental conditions. The downside? Well, it demands incredibly expensive lab instruments, highly skilled manpower, and advanced facilities. So while it's fast and reliable for sure, it is certainly not cheap or easy to deploy. Let's look at the results in Part 5, Real-World Crop Successes.
These aren't just theoretical concepts in a textbook, they are actively protecting our global food supply right now. Thanks to these specific molecular markers in lab applications, we've developed rice that resists bacterial blight and wheat that fights off rust. We've even created maize that can tolerate severe drought and run comprehensive improvement programs for cotton. The food we eat and the fibers we wear are quite literally stronger and more reliable today because of this highly precise lab work.
Which brings us to our final section, Part 6, the future of genetics. Just look at this incredible quote from our source material. Combined with genomics and AI, molecular markers have the potential to completely transform agriculture in the future. I mean, think about that. We are entering an era where the hyper-precise DNA tracking of molecular markers is merging with the processing power of artificial intelligence. This isn't just a minor upgrade, folks. It's a total revolution in farming that's going to make our agricultural systems significantly faster, more accurate, and much more resilient.
Which leaves us with one final, fascinating question to ponder. How will this rapidly advancing technology change the actual food that ends up on your plate in the next 10 years? It really makes you wonder, doesn't it? If you enjoyed this explainer and want to keep learning about these amazing deep dives into complex topics, please hit that like button, share this with a fellow focused learner, and subscribe. Until next time, keep learning.